Let's talk about Watson and Crick DNA. I like to think of DNA as a ladder. DNA is a double helix, and this representation is untwisted DNA. DNA strands are anti-parallel in nature because they run in opposite directions. The 5' end aligns with the 3' end of another strand. The orange pipe cleaners represent the phosphate sugar backbone, and the beads in the middle represent nucleotides and base pairing. A's match with T's and require 2 hydrogen bonding in the middle, and C's match with G's, which require 3 hydrogen bonds in the middle. Now that I've twisted this DNA, we can determine handedness. DNA is right-handed because it is twisted up and to the right. Let's take a look at the sugar phosphate backbone. Specifically, let's look at the sugar. The first prime carbon attaches to the base. Second prime carbon distinguishes DNA from RNA, and DNA and H will be attached here, and an RNA and OH. 5' carbon has a phosphate, and the 3' carbon bonds next to the 5' phosphate. I next want to look at major and minor grooves. Here you will see a major groove, and here you will see a minor groove. Major and minor grooves allow for protein placement. They are distinguished by the backbones being further apart in the major groove and closer together in the minor groove. Let's take a look at stabilizing factors in DNA. The hydrophobic interactions are maximized by base stacking. And base stacking is where purines and pyrimidines on both strands are stacked and help stabilize the double helix by van der Waals interactions. A's and G's are purines, and T's and C's are pyrimidines. I also would like to note that in the middle of DNA, the nature is hydrophobic, and on the outside backbones of DNA, it's hydrophilic. Let's take note that DNA is negatively charged because of the phosphate backbone. This is because the phosphate is negatively charged. So what happens to the melting point of DNA when there's an increase in salt concentration? The melting point is very reliant on salt concentration, so low salt will make DNA melt at a lower temperature than high salt concentration. So in conclusion, higher salt concentration means that DNA will melt at a higher melting point. The salt shields the negative charges on each of the phosphates on the phosphate backbone, and this destabilizes the DNA.